I'm John Chisholm, chair of the MIT Club of Northern California, and we are in for a special treat tonight. Our honored guest and speaker, Dr. Robert S. Langer, professor of chemical engineering, is one of only 13 institute professors, MIT's highest honor awarded to a faculty member. Dr. Langer works at the interface of biotechnology and materials science. A major focus is his study and development of polymers that deliver drugs, particularly genetically engineered proteins, continuously and at controlled rates for prolonged periods of time. He has written over 900 articles and has nearly 550 issued or pending patents worldwide, which have been licensed to over 180 companies worldwide. He is not only a prolific inventor, but also a trusted advisor and administrator. He chaired the Food and Drug Administration's Science Board, its highest advisory board, from 1999 to 2002. He has served at various times on 15 boards of directors and 30 scientific advisory boards, including those of Wyeth and Warner Lambert, and has received over 150 major awards, including the Charles Stark Draper Award from the National Academy of Engineering. This is perhaps the world's most prestigious engineering award, considered by many to be the equivalent of the Nobel Prize for Engineering. Both Forbes and Discover magazines have named Dr. Langer as one of the 20 or 25 most important individuals in biotechnology in the world. Time magazine and CNN named Dr. Langer as one of the 100 most important Americans and one of the 18 top people in science or medicine in America. Parade magazine selected Dr. Langer as one of 6, quote, heroes whose research may save your life, unquote. I could go on and on. Bob received his bachelor's degree from Cornell University in 1970 and his Doctor of Science from MIT in 1974, both in chemical engineering. We are honored that he will be speaking tonight on biomaterials and how they will change our lives. We are honored to be in his presence tonight. Would you please welcome Dr. Robert S. Langer.
Thanks so much. Thank you. Where's the, thank you. I just wondering if somebody could grab me some water. Thank you very much. It's a great honor for me to be here tonight and to be able to speak to you. I thought what I'd do, let me just pull this down, maybe I'll walk around, is I thought I'd tell you a little bit about actually my own career, how I got involved in this area, and then take you through some of the work we've done starting when I started at MIT all the way up to what we're doing right now. So my background, I finished my doctorate in 1974 at MIT. And I was in chemical engineering. And the major thing that almost all my classmates did in 1974 was actually go into the chemical or oil industry. That's what most chemical engineers did. In fact, what happened was that for those of you that are old enough to remember, in 1973 or 1974, there was an energy crisis. Thank you very much. And it was actually even a little different than the one we see today. The energy crisis in 1973 and 1974, you not only paid more money for your gas, but you actually had to wait in line for 2 hours to get gas. So, I mean, to me, that was actually a lot worse. The consequence of that is that if you were a chemical engineer, you would get a lot of job offers from the oil industry. And in fact, I actually got, this is true, I got 4 job offers from Exxon alone. And I wasn't that good. But the fact I got offers from Shell, all these places would offer me jobs like that. And I remember going to Exxon in Baton Rouge, Louisiana for one of my trips. It's funny, I was 24 years old at the time. I'd never been west of Buffalo, up till that time in my life. So I actually got to see the country. And I remember this trip to Baton Rouge, and I was at Exxon, and one of the scientists there said, if you could increase the yield of oil by 0.1%, he said, that would be worth billions of dollars. He said, isn't that wonderful? And I thought to myself as I flew back to Boston that night that I just didn't want to do that. And I was very interested in education. I had actually helped start a school for poor children in Cambridge and developed some new chemistry curricula, and I thought to myself, I'd really like to teach education. And I actually applied to a lot of schools, in fact, not even particularly good schools, like Loyola of Kansas and stuff like that, where they had offers open for teaching education. And actually, none of them wrote me back. And the thing is that, what I watched is people sort of pigeonhole you. You have a certain type of background, and that's what you get hired for. The next thing I thought about after none of them wrote me back was I also was interested in doing something with medicine. I hadn't taken any biology courses, but I always thought that might be an interesting and useful thing to do. So I applied to a lot of hospitals and medical schools. They didn't write me back either. Finally, one of the postdocs in my laboratory at that time mentioned to me, he said, Bob, there's this clinician named Dr. Judah Folkman at Harvard Medical School. He said, sometimes he takes unusual people. And I wrote to Dr. Folkman, and he called me up, and he told me what he was doing. And what he was doing in 1974 is he had this idea that if you could stop blood vessels, perhaps you could stop cancer. This was the idea that he would call angiogenesis. And he asked me if I could isolate what would hopefully become the very first substance that could stop blood vessels from growing. And one of the problems when you look at the history of biology and medicine, whenever you get into a new area, is one of the key problems is having an assay to study that. And that was a huge problem for studying substances that could stop blood vessels. And actually our idea was that we were going to actually look at cartilage, which is in your nose or your knee, and we thought cartilage actually didn't have blood vessels. So maybe we could isolate something out of cartilage, and it's mostly made of large molecules, peptides, proteins. We could isolate something out of cartilage that could stop blood vessels. We thought of the following assay. The eye of a rabbit. Rabbits have big eyes, and in their corneas, there's no blood vessels. But it turns out if you put a tumor in the eye, I'll show you pictures a little later, but if you put a tumor in the eye, it will cause blood vessels to grow over about a month or two period. And so we thought if we could then put possible inhibitors in the eye along with it, that might stop it and we would have an assay. The problem was whenever we try to make eye drops or anything else, the stuff just diffused away. So we thought what we'll have to do is try to come up with what we call controlled release system, drug delivery system for these large molecules. So that was 1974. It was kind of basic research. And what I could not have anticipated was that a few years later, because of some work out here, a whole new area would emerge, biotechnology, genetic engineering. And for the first time, it actually became possible to make large molecules. But large molecules face very serious delivery challenges. So it turned out the basic work we were doing, and I'll get back to the angiogenesis story a little later, but turned out to have implications that I could never have foreseen. Because if you tried to take any of these large molecules by swallowing them or put them in a patch, they're just too big. And then if you inject them in the body, they've got these really short lifetimes. So if you want to deliver any of these in a chronic basis, what you'd have to do is have a way to deliver it in an unaltered form and yet protect it from harm. But the conventional wisdom at that time was that you couldn't do this. So if you look in the literature in the 1970s, you often see quotes like this one. I'll read you this one, describing this then embryonic field. It said the agent to be released is a small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example proteins, could be released because of their extremely small permeation rates through polymers. Actually, this quote was very helpful to us in getting some of those early patents. It's actually true, you know. I mean, it's a long story, but in the end, we actually showed the patent examiner this quote, and he said, you know, I didn't realize that. I will allow this patent if you get the people who wrote the quote to write affidavits that they really wrote it. And he allowed the patent. I mean, I have a whole lecture on that. At any rate, what I did is I actually spent several years in the laboratory experimenting with different techniques. And I actually discovered over 200 different ways to get this to not work. But finally, I found one way to get it to work. What I found is that if I could take certain types of plastics, certain types of polymers, dissolve them in certain solvents, organic solvents, add the molecules to them, usually at a low temperature, dry them off slowly, I could make little microspheres like this. Here's one cut in half. And we published in the British journal Nature that you could use this approach to deliver molecules of almost any size for, this is over 100 days. Later on, we showed, using some engineering approaches, we even modeled how you could get constant release. And then, experimentally, we were able to get release of different molecules at steady rates, in this case for over 50 days. So I thought I'd tell you a story. You know, as one of the things that you have to do as a professor is you have to give talks. And in and I didn't necessarily realize this, but in 1976, I got asked for the very first time to give a talk at a major scientific meeting. It was in Midland, Michigan. And actually, up till that time, I really had never given a significant talk before. The only major talk I'd given, or significant talk to me, that I'd given before then was in 8th grade. And it didn't go real well in 8th grade. What happened in 8th grade is I had to give a 1.5-minute speech. So I wrote it out the night before, and I stood in front of my parents' mirror for 4 hours, reciting it over and over again. So finally, when I was 13 years old, and I got up in front of my 8th grade class, and I didn't bring the sheet of paper up that I'd written it on, and I got up in front of my 8th grade class, and I kind of like recited it, because I had memorized it, I thought, for about 1 minute and 2 seconds. And all of a sudden, I could not remember the next word. And I stood up in front of my 8th grade class for another minute, just like this, saying absolutely nothing, until my 8th grade teacher told me to sit down and gave me a not very good grade. And so that was always in the back of my mind whenever I had to give a talk. I did everything I could to avoid it. Well, now, I'll move back to 1976 when I had to give this talk. So this time I was really concerned about it. I knew that this would be important career-wise. So I actually stopped working about 2 weeks in advance of the talk, and I kept reciting the talk over and over again into a tape recorder. It was before VCRs or anything like that. So finally the day comes, 31 years ago, I'm giving this talk. You saw I was quite a bit younger then. I had more hair, I was darker. And I'm giving this talk to this very distinguished audience of polymer engineers and chemists, and all older than me. And when I got done with the talk, I actually thought I did all right. This time I didn't forget what I was going to say. I didn't stammer too much. So I thought when I got done with the talk that these scientists, these elder scientists, being nice people, would want to encourage me, this young guy. And when I got done, I stepped off the podium, and a whole bunch of them came up to me, and they said, "We don't believe anything you just said." It was kind of my introduction to how scientists sometimes treat other scientists. A point I will come back to later. About 3 years later, though, 1979, people began to reproduce what we did. And then the question shifted to how could this possibly happen? What's the mechanism? So what we did, I had a graduate student at MIT named Rajan Bawa. And what we did is we took techniques that were normally used in pathology, and we cut thin sections using a microtome, a pathology technique, to visualize what was actually happening. And what you're looking at here is a 5-micron thin section of one of the polymers, ethylene vinyl acetate. There's no drug in it. And if you did a permeability study, you'd find molecules 300 molecular grade or greater can't diffuse from one side to the other. So how do the molecules get through? Well, now I'm going to show you another section. The only difference is we put a reddish protein in, myoglobin, and you see a phase separation. And what you see is before any release has taken place in this thin section is the polymers this white and the proteins this red. Now, if you release this, say for a year, and then you cut a thin section, what you see is left behind where the drug was are these pores. You didn't see them 2 slides ago. So the fact is these pores were made by the way we created these systems. And and it turns out, if you do more microscopy, more imaging, what you see is these pores are interconnected, and there are many tight constrictions between them. They're also incredibly winding and tortuous, so it takes a really long time to get through them. The way I always explain this to people is it's kind of like driving a car through Boston. And it turns out that by controlling the porosity, you can make the delivery anywhere from a day to over 3 years, or almost any time in between. In fact, the biggest problem that I have found as an MIT professor in getting really long-term release is actually not the design of these systems, but rather my ability to find a PhD student who will do the experiment long enough. And I was very fortunate in the 1980s. I had a very good graduate student named Mark Saltzman. He actually did this experiment for over 3 years, kept coming out. And then one day, Mark said he wanted to talk to me. I said, sure. I said, what would you like to go over? He said, Bob, I've been thinking. He said, what will I do if this experiment fails? He said, do I have to repeat it? So we stopped at 3 years and 2 months and 5 days. But I've always thought with the right student, we could go 6 or 7 years. And actually, I always tell people it paid off. Mark today is the inaugural chairman of biomedical engineering at Yale University. He's the Gauzietta professor there, and they're just thrilled to have him at Yale University. So it's worthwhile doing these kinds of experiments. Now, I mentioned to you in the beginning of the talk that the main reason that was initially driving me to do this was this angiogenesis problem. There were no assays for angiogenesis molecules. And so now that we had this ability, we actually were able to, for the first time, to see if we could isolate angiogenic stimulators and inhibitors. So we developed these assays. Here's the rabbit eye assay. Here's the polymer. Here's the cornea. And notice there's no blood vessels. This is time 0. But if you put a growth factor in, like FGF or VEGF, various growth factors, what will happen is 17 days later, it looks like this. And this would provide a basis for all future work for isolating these angiogenic stimulators. For an inhibitor, the assay would be you'd put the tumor in, you'd have a slow-release polymer, and you'd actually see if you could stop blood vessels from growing. And in fact, using this approach, we isolated the very first angiogenesis inhibitor. It was published in Science. It was initially from cartilage. If you didn't have the inhibitor in, this is about 4 weeks out, you see a sheet of blood vessels growing throughout the eye. They start here, grow throughout the eye, over the polymer. You can't even see the polymer, towards the tumor. If I waited 1 more week, this would grow out of its own orbit. It would be, you wouldn't want to see it. It would be quite gross, but it would be 3-dimensional. In contrast, if you put the inhibitor in the polymer, notice what happens. The vessels avoid it, and they're not nearly as high. And this would provide a way, not only did it prove that you could actually have angiogenic inhibitors, it provided a way to isolate all future angiogenic inhibitors. It took a long time from this study we published in Science, I think in 1976, for the first angiogenic inhibitors to get approved. In fact, the first one was approved out here at Genentech. But basically what's happened is 28 years after we did this, all of a sudden you see an enormous number of approvals. Avastin from Genentech approved for colorectal cancer in 2004. Tarceva approved for lung cancer in 2004. Macugen, this is approved for macular degeneration. That's a leading cause of blindness, approved in 2004. Nexavar approved for kidney cancer in 2005. Revlimid approved for another type of cancer in 2005. Sutin, another angiogenic inhibitor, approved for gastric and kidney cancer in 2006. Lucentis, again from Genentech out here, approved for macular degeneration, actually reversing it in 2006. And we continue to see these now approvals. Again, a lot of times in medical research, things take a long time, but it's been very rewarding to see this happen. Now, we also wanted to see if we could use this approach maybe in the nearer term to release other molecules. And what we did is we took as a model insulin. This was Larry Brown's PhD thesis, putting insulin, 6,000 molecular weight in pellets, putting it in diabetic rats, showing the first sort of systemic in vivo proof of principle that you could deliver a protein for over a month. This is actually over 100 days. And what I did is actually, it turned out that for 10 years, we'd publish papers and people, would read them and nobody really cared. Finally, actually, a Japanese company came to our lab, gave us a grant. We taught them how to do it. Actually got involved in starting a company myself. Today, that company's Alchemies, which now develops these things. And there's a whole bunch now of various controlled release systems based on these technologies. So this has actually been a very successful one. It's for releasing luteinizing hormone, releasing hormone analogs. You absolutely have to use this kind of controlled release system. This is a peptide. If you tried to take it orally or nasally, you get nothing in. If you inject it, it's destroyed in a couple of minutes. But now, originally they lasted for a month. Now these injections actually last for 4 months. And there are various versions of it. Also, other diseases where this has made an impact are mental health diseases. If you... by the way, for the economy, this was a couple $1 billion a year product. If somebody, like I don't know how, just to give another example, how many of you here saw A Beautiful Mind with Russell Crowe? Quite a few, everybody. So you might know he had schizophrenia, so he'd see people. And the way they treated him was shock treatments. Today, though, if he had it, what they might give him is this shown here, Risperdal Consta. What happens is now you can take a drug like this, put it in a controlled release system. In other words, compliance is a real big issue for people with mental health problems. This is already a $1 billion a year drug, too. They give injections once every couple weeks. This just got approved recently for treating alcoholism, once-a-month injection. And there are many, many others in various stages of clinical trials. So today, it's certainly possible to release large molecules, to release them at constant rates or even decreasing rates. But you always like to do better. And about 13 years ago, I was watching a television show on PBS. The Red Sox weren't doing that well that year, so I was... and I was watching how they made microchips in the computer industry. And as I was watching it, I thought to myself, boy, wouldn't this be a great way to make a drug delivery system? Now, if you spent 30 years of your life working on drug delivery systems, maybe any TV show you saw, you might think that. So as I saw it, I thought to myself, well, could we make a little microchip into a drug delivery system? And I called up Michael Sema, who's a professor at MIT and does ceramics processing. And I talked to him about this, and we had this idea of making these chips. Here's a cutaway. You have a backing membrane. You have little wells you build in, all, by the way, using techniques in microelectronics processing, but never before had they been used in the pharmaceutical area. And then we covered them with little gold caps. And the idea is you could have these wells, and in these wells you could put different drugs or different doses of the same drug. You could literally have a pharmacy on a chip if you wanted. So this was our idea. We got a very good graduate student named John Santini to do this for his thesis. And we actually made some of these chips. I'll show you an early chip. John now has a company. He actually gets mad at me when I show this slide, because this was an early chip he made for his PhD thesis. Now he has a company that makes these chips, and they have 400 wells on them and all kinds of bells and whistles. But here's an early chip, 34 wells in the top, 34 in the bottom. Here's a United States dime. And by the way, these chips can be made in all kinds of ways. You can make big chips, little chips. You can make 50% of the space active drug if you want. You can make injectable chips. You can make fully degradable chips. We've had students do all of these things. Let me show you how they work, though. A standard chip might have gold in it, though it doesn't have to. And here's an example of what John did, also published in Nature. You have a well covered with gold, and underneath it's a drug. And what you can do is apply 1 volt in the presence of a small amount of chloride ion, which you'd have in your body, and in 10 seconds, here's what happens. The gold comes off. You can actually take 16,000 times this amount of gold, put it in animals. Jim Anderson, an outstanding pathologist at Case Western, has done this working with us. You see no harm. So the idea then is you could do remote control delivery. And here's an example of that John did, where he put different amounts of drug in different wells and triggered release at a day and a half, 2.5, 4.5, 5.5 days. If you wanted to do the pharmacy on the chip idea, here he triggered release of 1 drug at 25 hours, another at 37, another at 50, and another at 60. It's purely arbitrary. He could have done it whenever he wanted. Now, in the long run, what we believe will happen, in fact, John and his group at Microchips published a paper in Nature Biotechnology last year showing that you could actually have a chip, you could even put it in dogs, and you could, by telemetry, remote control, even like a wristwatch, you could trigger release over and over and over again whenever you want. It could be pre-programmed, could be turned on, and even a fairly large molecule like a peptide could come out over a 9-month period. No harm to the animals. In the longer run, what we believe is that what you could do is that you could actually also build sensors on these chips. We're doing that as well. Have the sensors sense things in the body and then tell the chip in turn how much drug to deliver. So you could actually someday envision a chip, a smart chip that could sense things in the body, tell the chip how much to deliver. And in addition, one more thing it could do, as I've gotten older, one of the things I've noticed is, my memory's not as good. And one of the other big problems sometimes is medical record keeping. So whenever the chip gives a drug, that information about how much drug got delivered and when could actually be transmitted to a computer at your house, the doctor's office, or a hospital, so there could be a permanent record of whatever you've done. So this is hopefully something that will happen sometime in the future. Now, so far what I've done is I've talked to you about how we've engineered materials to do things they've never done before. But we've used materials that already exist in nature. I want to go over another thing that we've done where we actually designed brand new materials. When I graduated from MIT, as I mentioned to you, I went to Boston Children's Hospital. That was considered a very unusual thing at the time. One of the things that I was curious about when I went there was how do materials find their way into medicine? And I thought naively that it must have been older chemical engineers or chemists or material scientists that were responsible for doing this. But when I looked into this in the hospital, I found that was almost never true. Almost always, the driving force for bringing materials into medicine were medical doctors. And what they always wanted to do, because they urgently, and that's important, they urgently wanted to solve a problem, is they went to their house and they asked the question, what object in my house kind of resembles the organ or tissue we want to fix? And then they'd use it in a person. And I know people might think this is funny, but it's also true. So let me actually just show you the next slide. And this is true. So I'll just tell you a few examples. In 1967, some of the clinicians at the NIH wanted to make an artificial heart. And they said, well, what object has a good flex life like a heart? They said, a lady's girdle. They said, what's it made out of? It's made out of a polyetherurethane. So they decided in 1967 to make the artificial heart out of the lady's girdle material. But the thing is, as you people who work in medicine know, once you start down that path from a regulatory standpoint, it's not so easy to change. So today, 2007, 40 years later, that's still what it's made out of. Why has the artificial heart not worked so well? Well, you don't need to read Science Magazine or New England Journal of Medicine. You could read the San Francisco Chronicle or the Boston Globe, and what it would tell you is why it's not worked well, and you can just read this, is that when blood hits the artificial heart, it forms a clot sometimes. That clot goes to the patient's brain, and they get a stroke, and they may die. And yet, if you think about it, something that was designed to be a lady's girdle, probably isn't the optimal material to put in contact with blood. And this problem pervades all of medicine. Dialysis tubing with sausage casing. Vascular graft, that's an artificial blood vessel. That was a surgeon in Texas going to a clothes store to see what he could sew well with. Breast implants. One was a lubricant. The other actually a mattress stuffing. And I realize people think it's funny, but every one of these is true, and I could actually make this list longer. So I started seeing this, and I thought to myself, what they teach us at MIT is they teach us engineering design. So rather than do this approach of taking stuff in your house, I thought, what if we ask the question, what do we really want in a biomaterial from an engineering standpoint, from a chemistry standpoint, from a biology standpoint, and then could we synthesize it from first principles? So we picked an example. And the example we picked were materials. So when we originally started, the only materials approved by FDA were polyester sutures. And they dissolve like this. They start out like this, they get spongy, and then they fall apart. The difficulty with this is that when they do this, they could burst open like this. And if you had a toxic drug like a cancer drug or insulin, it could all dump out. So we said from an engineering standpoint, what you want is this, surface erosion. And we went through a very detailed engineering design approach to get there. we asked what types of do you want it to be degraded by enzymes or water? We said everybody in this room has different enzyme levels, but you all have excess water. So we said we'll make the polymer degrade by water. Then we went to figure out what were the right monomers, what were the right chemical bonds, and so forth. And I won't go into all that unless people want me to in the question period. But in the end, what we did is we arrived at this particular polymer. We even had toxicologists helping us. In fact, one of the people who helped us a lot at the time at MIT was Michael Marletta, who was one of my close colleagues at MIT. Now he's actually chairman of chemistry at Berkeley, and UC Berkeley. But here's one of the polymers we developed. It's a polyanhydride, actually. The anhydride bond would be here with two monomers. This is hydrophobic, and this slightly less so. And what was exciting is that if you make different levels of those two monomers, 0% of the one, 15%, 55%, 79%, look at the different rates at which these dissolve. This will actually take 3 or 4 years for a millimeter-thick slab to dissolve, whereas this is all gone in 2 weeks. So you could simply dial in the ratios of these and make it last almost any length of time you want. So this gives you enormous power for targeting different medical applications. So let me give you a real example. In 1985, a young neurosurgeon named Henry Brem, just starting his career at Johns Hopkins, came to see me. He wanted to come up with a better way to treat brain cancer. Today, actually, Henry, because of the work I'm going to talk to you about in large part, is chief of neurosurgery at Johns Hopkins. But here's the most common form of brain cancer, glioblastoma multiforme, uniformly fatal. Regardless of how you treat the patient, the mean lifespan is less than a year. The drug that is normally given in this case is this one, BCNU. But normally, of course, it's given systemically. Intravenously, it travels throughout the whole body, causing serious side effects to the liver, kidney, and spleen, as do many chemotherapy drugs. So we had an idea, and that idea would become local polymer chemotherapy. We asked, rather than give it throughout the whole body, what if the neurosurgeon, like Dr. Brem, would operate on the patient, and then before he closed the patients up, could he line the surgical cavity with the polymer containing this drug? Normally, the drug has a 12-minute lifetime, but if you put it in the polymer, it could be protected and last as long as the polymer lasted. So what the neurosurgeons wanted was a polymer that would be degradable so it wouldn't accumulate in the brain, that would have the surface degrading properties so it wouldn't dump the drug. And from animal studies they did at Johns Hopkins, they wanted it to last for a month. All that could be done with these polymers. So the polymer protects it from degradation, but perhaps most importantly, we get local chemotherapy, high concentrations in the brain where you want, and almost no concentration in the rest of the body where it could cause harm. Now, one of the things that happens is, see, if you're a professor at MIT or really anywhere, is if you have ideas like new polymers or new kinds of therapies, you have to raise money. And the way we raise money at MIT is we write grants. In my case, the most common way I write them is to the National Institutes of Health. And then what they do is they get study sections, which are maybe 20 people from other universities, to review the grants. And that's what happened. And actually, the grants didn't do very well. I entitled this slide, "This Approach Will Not Work Because." So in 1981, when we first proposed this idea, the chemists looked at it and they said the polymers could never be synthesized. But I had a very good graduate student at the time named Howie Rosen. Howie would eventually become president of the Alza Corporation out here, now senior vice president at Gilead, also out here, was elected to the National Academy of Engineering a few years ago. And he synthesized them. So then we sent it back again to the NIH, and the reviewer said, well, that's nice, but, anhydrides, they're really reactive bonds. They'll react with whatever drug you put in. But I had another couple postdocs, Bob Linhart, who's now Constellation Professor at RPI, and Cam Leung, who's the James Duke Professor of Bioengineering at Duke University. And they figured out ways, using salts and other approaches, to make sure there would be no reaction. So we sent it back again. By the way, they're not real fast at NIH. So we sent it back again, and the reviewer said, well, that's nice, but it won't work. The polymers are low molecular weight. They're fragile. So I had another couple postdocs, Avi Dome, who's now chairman of medicinal chemistry at Hebrew University, and Edith Mathiowicz, who's now full professor of bioengineering at Brown University. And using the right catalysts and reaction conditions, they made them really high molecular weight, really strong. So they weren't fragile anymore. Sent it back again, and the reviewer said, well, new materials, they're certainly going to be toxic. I had another graduate student, Cato Lorenzen. Cato is now university professor and chairman of orthopedic surgery at the University of Virginia. He was elected to the Institute of Medicine of the National Academy of Sciences 2 years ago, and he showed, actually supporting Marletta's early theories, that these, in fact, were quite safe. Anyhow, this kept going on and on until 1996, when the FDA approved it. It was actually the first time in over 20 years they approved a new treatment for brain cancer, and the first time they ever approved this idea of a polymer-based local chemotherapy. Now, one of the things you can probably tell from the way I'm speaking is that I'm really very, very proud of how well our MIT graduate students and postdocs have done. They've become chairmen of major corporations, chairpeople of major departments. They hold endowed chairs. Whereas the reviewers at the other schools, they haven't done so well. Now, I'd actually like to show you what this operation looks like, but I'm serious about this. But if anybody's squeamish and doesn't like the sight of blood, you shouldn't look. But let me just show you. These are going to be human brains, and little wafers going into the brain. Usually, they put 7 or 8 in, close up the brain. I hope people saw it. Actually, the reason I did that, it's very hard when you give a talk to get good advice. But several years ago, I was actually giving a lecture at MIT, and my wife came to the talk. And at the end of the talk, I said to her, Laura, I said, what did you think of the talk? And she said, well, Bob, the talk was okay. That's actually very high praise. But she said, you had those 2 bloody slides on for nearly 10 minutes, telling the audience every detail of the operation. She said, I don't know if you were looking, but all those poor engineers, they were turning green. So ever after, I did just what I did tonight. But I do want to tell you the sequel to this, was that about 2 years after that, I got asked to give a dinner speech. But this, unlike tonight, was to a group of neurosurgeons. And I told them the same story. It was actually at the Boston Aquarium. I told them the same story. And at the end of it, they said, those 2 bloody slides you showed, they said, those were fine. They said, no problem. They said, but those chemical formulas... to show those to a neurosurgeon, So now if I show the neurosurgeons, I show those chemical formula slides really quick. At anyhow, to show you a little bit of the clinical data, this is from what's called the phase 3 trial right before FDA approval. You see 63% survival in the treated group, 19% in the control, which is the best conventional treatment. At the end of 2 years, 31% in the treated group, 6% in the control group. And this got approved originally for recurrent brain cancer, then more recently for primary brain cancer. Now people are using this kind of approach that recently got approved for a spinal cancer, clinical trials for other cancers. But the area where this concept has had the greatest impact actually turns out not to be cancer at all, but in interventional cardiology. And in fact, I'm sure people read about this, but that now one way, if somebody has heart disease, they actually put these stents in patients' blood vessels to keep them open. But a lot of times they clot off, or you get restenosis because of cellular proliferation. In fact, one of my other graduate students, Elazar Edelman, who's now a professor at MIT and Harvard, actually has done a lot of work on this. And basically, you take the same idea. You take a toxic cancer drug like Taxol, put it in a polymer, coat the stent, locally deliver it. And these have had very substantial effects on preventing restenosis. And actually, even though the first ones were approved in, I think, 2003, already the market's something like $4-5 billion a year. Now, so these are some examples of materials. I thought I'd give you one other one just to tell you how I got, I remember also thinking at one time about this area of minimally invasive surgery. And it used to be that if you had an operation, maybe 20, 25 years ago, let's say you had your gallbladder out. What they used to do is they'd make a big incision in you, and they'd take the gallbladder out. Now, what they do is they make a tiny incision, and by the way, when they took the gallbladder out, because they made such a big incision, you'd be in the hospital for a week or two. You wouldn't be back to work for several months because they made a big wound in you. Now, they make these little incisions. They put these scopes in it, and on a TV screen, you can see everything. And you can take the gallbladder out through that little hole. You're out of the hospital in less than a day because they made a tiny wound in you. You're back to work within less than a week, again, because they've made such a small wound. So it's really had a major effect on hospital care and so forth. So I started thinking, you know, that's really neat that you can do that, you know, that you can take things out. But could you ever put things in, like bulky medical devices, through those little holes? Now, that might seem like science fiction. How could you do this? So I had this idea that maybe what we could do is make a series of what I'll call shape memory polymers that are biodegradable, biocompatible, that you could start out at room temperature like a string, and then you could put them through these holes. And you could program them either by temperature or light. We've actually published one paper in Science, one in Nature, one, but I'll pick the temperature one as an example. But you could do it either way. With light, you do a fiber optic. But the idea is that we could make a series of what I'll call shape memory polymers that at room temperature could be like a string, and in body temperature, different temperature, could be whatever shape we programmed it to be. Might sound like science fiction, but actually I had a very good postdoc named Andreas Lendlein. And for the chemists in the audience, what we did is we made a series of what we call phase-segregated multi-block copolymers. And the idea is that one block controls the shape at room temperature, the other at body temperature, if you use temperature as a trigger. And we build in one as a series of crosslinks that will melt, say, at 30 degrees C. So let me just show you a little of what we did. I'm going to show you a video. And what I hope you'll see is a string at room temperature in air. And then we're going to drop it into body temperature water. Hopefully it'll change to a coil. So here's the string at room temperature air. We're going to drop it into body temperature water. And that's what we programmed it to. By the way, that could have been programmed to anything, to any shape. It's just a question of what you designed it to. Let me give you a second example. Let's say you had a wound. So if you had a wound on your face or your arm, you tie a surgical knot. And you could do that. I could do that. You wouldn't want me to do it, but I could do it. It's outside the body. But what about if you had a wound in your stomach or your lung? How could you tie the knot then? So we thought, what if we could make the knots tie themselves? What if we looped in a loose knot like a lasso at room temperature, and then when it gets to body temperature, it just tightens? So let me show you a second video. So here you see at room temperature the loose knot. We're going to drop it into body temperature water. Hopefully it'll tighten. And these are real-time, real-temperature things. So it's early stage, but gives you a view of what the future might be. Let me go over the last topic, which is, could we not only use materials to do these kinds of things, deliver drugs, make new devices, but could we someday, there's so many patients, it turns out, that have problems that you can't fix by drugs or devices. And let me just give you an example. I'm going to show you a patient on the next slide that's dying of liver failure. Here I work very closely with Jay Vacanti, who's head of pediatric surgery at Mass General Hospital. And this little boy is a patient of Jay's. And he's dying of liver failure. And the only way a child like that lives is somebody else dies. And then the surgeon will take the liver and do a transplant. But the problem, there's a lot of problems, but the biggest problem is donor shortage. Not enough people die. And so, which is good, but the thing is that you have this donor shortage problem. So we wondered many years ago whether we could solve this problem by taking cells from the patient themselves or a close relative. And if we could, these are bone, cartilage, liver, intestine, urothelial cell. It turns out our thought was as follows. If you take these isolated dissociated cells and inject them at random, nothing happens. But cells are smart. If you take cells and put them close enough together, they actually can form new structures. A group out here at Berkeley has shown that you can take mammary epithelial cells, put them close enough together in vitro, they'll actually make acini and make milk. So our thought was that we could make a three-dimensional polymer scaffold where the cells could be close enough together to really communicate and form these structures. Figure out the right way to grow it, and then ultimately put it in the patient. So this was the idea. And let me just show you a scanning electron micrograph of one of these polymer systems. And here there are fibers, and you put liver cells on, but it could be anything. Now, and also one of the beauties of this is that you could really make these in any kind of shapes. So Prasad Shastri, one of my postdocs, actually worked out a way to make polymer scaffolds in virtually any shape that are about 98% porous, so that you could design these. And just to pick an example, like what if you wanted a new nose? Let's say 30 years from now, somebody goes to the plastic surgeon, they want a new nose. I believe what could happen 30, 40, 50 years from now is if somebody wants a new nose, they go to the plastic surgeon, and you'll be able to use CAD/CAM techniques like computer-aided design to make any kind of nose you want. So here's the polymer. Prasad kind of made a regular nose here. But you might have on a computer screen, say somebody wants an upturned nose. You know, you take a little off. What if somebody wanted a hook nose? I mean, they probably wouldn't. But if they did, you know, we just give them a little bit more here. And then you take their own cells. In fact, we're not doing this in noses. We're doing this in other areas, which I'll show you. Take the patient's own cells, maybe from a few cells from the ear, put them on the scaffold, make a new nose. Well, let me give you, to end the talk, 3 examples of some of the tissues that we've worked on. We've actually worked on over 20, but I thought I would just give you a couple of examples. What if you wanted to make new cartilage? Here we work with Chuck Vacanti, and as a model, we actually did this plastic surgery. We redid this guy's skull. And it doesn't reject because it's his own cells. It's a nude mice. We redid this guy's cheek. If you open these animals up and look at them, you get pure white glistening cartilage like this. And if you look at it histologically, you have all the characteristics of regular cartilage. It turns out, however, that the cartilage is not of high enough quality yet for weight-bearing problems, so we're continuing to work on understanding that. It is, however, of high enough quality for a variety of cosmetic applications. So Chuck also sees patients that don't have ears, where the ears are sliced off. So here, Linda Griffith, who was one of my postdocs, she's also now a professor at MIT, made a new ear. This is a scaffold in the form of a human ear. Here's a close-up of just a small portion of it, with the polymer fibers, the cartilage cells, the matrix it's making. Over time, the polymers will disappear, and you'll have all cells in matrix. And Chuck actually does not yet put this on humans. He has put it on rabbits. And here's the rabbit with the human ear. Now, I know people might ask, well, why are we doing this to rabbits? But the reason we do it to rabbits is because we want to know what's safe for patients. And so what's been done by some of my clinical collaborators is various physician-sponsored INDs. Here's a little boy at the time, 12 years old. Notice he has no chest covering his heart. But like other 12-year-olds, he likes to play baseball. But if he ever got hit in the chest with a baseball, he could die. So we actually made a scaffold for him, like a chest, took his own cells, Jay operated on him, and gave him a brand new chest. And that was 10 years ago. He's still doing fine. There have been other examples of this as well. Another example could be new skin. Here we licensed this technology to a company, and they made new skin for burn victims. This is now FDA approved. Here's a little boy, very badly burned, but you could take the product, which is a polymer scaffold with a neonatal skin fibroblast, cryopreserve it. You could put it in the child at the time of injury, and it looks like this. But if you come back just 3 weeks later, he looks like this. If you come back 6 months later, he looks like this. Pretty much healed. And like I say, these have now been approved for burn victims and patients with diabetic ulcers. Let me end with one final example, which is still in the animal testing stage. But we also thought that we might be able to use this approach to maybe someday help people who are paralyzed. And Aaron Levick, who was one of my graduate students at MIT, now professor at Yale, worked with Ted Tang, who's a surgeon, and Evan Snyder, who's a neuronal stem cell expert. And Erin, for her thesis, designed a polymer scaffold really to mimic a spinal cord. Would have an outer portion to hopefully provide axonal guidance, and an inner portion where she would put neuronal stem cells. And what was done is to actually take a segment out of the spinal cord of different rats and then replace it with one of 4 things. 3 of them were controls. So you replace it with nothing, cells by themselves, or polymer by itself. But in the set of experiments, what was done, using 12 animals, we did about 50 animals overall, and followed them for over a year, actually. What we did is we would put the combination of the scaffold and the neuronal stem cells in. And what I'm going to show you is now 100 days out, first the mean of our control group, and then the mean of our experimental group. So here's a video, 100 days out. And what I want you to do is focus. These experiments that we did, we took out a section which makes the rat paraplegic. So this is 100 days after the operation, and he's dragging his feet. Also notice how they splay the paws. There's actually a scoring system that you can use to quantitate this, a BBB score. But you get the general idea of what happens We'll just watch this a little longer. And this is pretty typical of the controls. Some did a little better, some did a little worse. Let me now turn to the mean of the experimental group, keeping in mind that rats are not humans and that we've got a long, long ways to go. And even in the rat study, it's not a cure. But what you'll see is a marked improvement. So in this final set of slides, or videos, here's the treated rat. This, again, is our mean. And I think there's a couple things to keep in mind. He's able to bear his own weight. And notice he's actually kind of heavy. But he is able to bear his own weight. He's a little bit clumsy, but he's able to bear his own weight. And we'll watch him walk a little bit. And so you can see the marked contrast to the... You notice the paws are splayed in a much more normal fashion than the last guy? My wife tells me not to leave that on too long either. Well, that's what I want to go over with you tonight. You know, if I look at what we've done, we've got a long, long ways to go. I mean, we've probably raised more questions than we've answered, which is good in some ways. We'll keep a lot of MIT graduate students and postdocs busy for years to come. But at the same time, we feel like we have, you know, if I go back now to over 30 years ago when we started, and people were very skeptical about whether you could do some of these things, what we are beginning to see is the dawn of new medical therapies, in a variety of areas, drug delivery, the beginning of tissue engineering. And it's certainly my hope that as we and many, many people throughout the world move forward in these areas of bioengineering, that this will just be the tip of the iceberg, and that there'll be many, many more types of products that can relieve suffering and prolong life. Thank you very, very much.